chaff falls away. This can be prohibitive when the number of reads is very large and when the proportion of the reads that represent what you want is really small. Your assembler then chokes if it's unhappy with you. You can also try to filter prior to assembly. One example would be to just blast everything and try to figure out which reads might be what you want and get rid of the other ones. I'm collaborating with some people on this and we were having some issues, so I wanted to develop a new approach, a very simple bioinformatic approach to try to filter reads based on whether they're likely to be assembled or not. And what we're doing is we're using K-mer composition, where we profile not just the targets of what we want, but profile likely contaminants. And by profiling both what you want to do and the contaminants, we think we can get better discrimination among the reads that we actually do bring in. This is based on K-mer composition, right? A K-mer is just a stretch of DNA K nucleotides long, and of course there are 4K possible K-mers of length K. So as the example, what we did is we started by getting 89 complete mycobacterium genomes from GenBank. These were all of the ones available at the time. Roughly half of them were from the Mycobacterium tuberculosis complex. That's the target group that we want. We're looking for things that cause tuberculosis. And then the other half were from the entire rest of the genus. So this is one fairly small clade within the much larger genus. Most of these were actually Mycobacterium tuberculosis, various different strains of them. For both groups, we calculated the K-mer composition for a range of K. In this case, K-mers were scored as just present, absent. No attempt was made to look at frequency, just does that K-mer exist in the group, yes or no? And I'll come back to that a little bit. So some very obvious and expected results before we actually try to profile anything is first, not surprisingly, that when K-mer length is short, let's say around 10, you find virtually every possible K-mer that there could be. In the data set, it's not quite 100%, but it's very close. But as K gets longer, you start getting only a tiny fraction of all the possible K-mers. That's not surprising, but it drops very rapidly from virtually everything you can find to almost nothing that you can find. If we actually look at specifically how these K-mers are distributed between our two groups, at short lengths, the vast majority of K-mers, 90%, are actually found in both groups, with very few that are unique to either group. But again, as K gets longer, the overlap drops very precipitously, almost down to close to 0, while the uniqueness goes up a bit. You'll note that there's a lot more uniqueness in the non-tuberculosis complex than in the tuberculosis complex. Again, that's to be expected because this represents a much broader taxonomic range, while this is just a small group of clade. I do want to point out that there actually is quite a bit of movement going on in this line here. It just looks flat when you plot it against the other bars. So looking at this, we're trying to figure out what would a good K be to use. There's a trade-off. The longer you make your K-mer length, it starts to take more time and memory, and it's sort of an exponential growth. So you don't necessarily want to do everything at super long Ks. We guessed that the optimal range would be somewhere in the 12 to 15 range. 12 is where these start to flip over. At 15, you're pretty much getting to these flatter parts of the curve. So then what we did is I developed a score, and the scoring system is very, very simple. It's just algebraic. There's no hidden Markov model. There's no Bayesian anything. This is just basically, if you take an unknown read and you look at all the K-mers in that read, and you ask, are the K-mers in the target group, are they in the contaminant group, are they in both, or are they in neither? So the 4 possibilities. And you just sort of algebraically add that up and rescale it. We end up with a score that ranges from 100 to negative 100. Basically, reads which look more like the desired target should have high positive scores. Oops. Reads which look most like the contaminants should have negative scores. Reads which are ambiguous should have scores about 50, and reads which are unknown would be about 0. To put this on a scale, things up here are completely unique to the target group in K-mer composition. Things down here are completely unique to the contaminants. Things that are common to both highly conserved regions should be around here. Unknown sequences would be there. I do want to point out that you can get these numbers through other combinations. So we could get a 0 if exactly half of your K-mers were unique to the target and exactly half were unique to the contaminant. This is deliberately asymmetric in the way it was set up, and you don't want to overinterpret these numbers. It's just a way of putting things on a scale that gives you some flexibility. The important part is that scores greater than 50 have to be at least look somewhat like uniquely to the target. Things that are positive at least have to have something in common with the target. So to sort of now actually apply this, we started with a sort of very simple simulation. Basically took known genomes, things that we knew were right, simulated pulling out little reads of average length. In this case, it's all 70 bases, which is about the average that we get for ancient DNA sequencing. With a K of 12, and then we actually took those and randomly induced SNPs and indels in them so that we would sort of put a little bit of error unexpected. With a K of 12, you can't really distinguish anything. The lines all bump up against each other. But as K gets larger and larger, we start to see separation. K of 13, there's still a lot of overlap between the non-tuberculosis complex and the slightly altered TB genomes. But at 14 and 15, we've got very nice separation. We should be able to tell them apart in principle. These were all done with fixed sequence length. This is scales though. This is where all the reads are 70 bases. This is where the reads randomly vary from 50 to 150, which is about the range we get out from most of the ancient DNA sequencing. And it's basically the same distribution. So this is nicely independent of the length of the reads. So then we want to see what happens if we use this on real data. So now this data comes from this was published a few years ago. This is from an ancient DNA study on pre-Columbian, these are Peruvian mummies, which had signs of tuberculosis, and they successfully extracted ancient tuberculosis from multiple mummies in this study. This is one of the samples. We've repeated this for all the samples. The results are very similar for all of them. I'm just going to demonstrate using this particular one. So these were all 19 million reads that they got out from this particular study. These are just the scores blind of all 19 million reads. We immediately see there are a minimum of two distributions here. We have a high group of very high positive scoring ones, and we have this big peak of slightly negative scoring ones. There's this extra weird thing right here. That's a score of exactly 50. That would be that conserved overlap, both groups sort of region. There's also this extra little peak right about 0. Maybe this is where something unknown's in there. We're not sure. The important thing is for this, you can actually just dump this on the assembler, give it a reference genome and say, let's assemble it and see what happens. So we did that, and then we separated everything that went up against the assembly to those reads that successfully assembled and those that didn't assemble. And then we looked at the score distributions of those two groups, and what we find is, in fact, the things that successfully assembled are overwhelmingly in this nice positive area. There is a tail, which I'll come back to in a moment. And the things that failed to align are, of course, this massive peak down here, but also quite a lot of positive stuff, which again I'm going to come back to. So superficially, it does seem like we can identify the things that are likely to align using this score. We can identify the things that are likely not to align using this score. So what actually would be the effect of using this as a filter on the assembly? So we just arbitrarily said, what happens if we get rid of everything whose score was less than 25? And we actually chose that before we ever plotted any of these histograms. We just said, let's say 25 is a filter, and just get rid of, take all 19 million reads, score them, just throw out all the ones whose score is less than 25, and then do an assembly with what's left and see what happens. So the first thing that happens is we go from almost 19 million sequences down to less than 7 million. That's before we assemble, and we put them in the assembler, and we find this: the number of mapped reads, the number of quality mapped reads, the number of unique mapped reads, the mean coverage, the percent of the reference with one-fold coverage are essentially, functionally unchanged. Basically, we got rid of 64% of the input to the assembler, but ended up with an assembly which is, more or less identical. And so this did work. It took a huge load off the assembler. The assembler worked a lot faster once you've done this filtering, but we didn't affect in a major way our assembly, which was really nice and what we wanted. Now 25 was completely arbitrary. If we go back to this graph and look at it, we can see that about 84% of the reads that successfully aligned had scores of 50 or greater. 97% were at that 25 mark, 99% of the reads that are going to assemble have scores greater than 10, and 99.5% had positive scores. I mean, if we set the filter to positive scores, we're still getting rid of a vast amount of the stuff that isn't going to do any good anyway. And so from a general standpoint in terms of pre-assembly filtering, it seems like this approach will work. It's very early in terms of its development. There are a lot of things we still do need to do. One of the things that's sort of interesting about it and that we want to look at more carefully is if we come back to these plots which I showed you before, is there's this long little tail here of things that did actually successfully assemble, including, I don't know if you can even see it, some very negative scoring reads. And I actually want to go back and look specifically at these negative scoring reads down here and figure out why are they aligning to the reference genome. What is there, are these just sort of false positives that are coming out, or is there something here that is important that's not being captured? The other big question is why is there this huge peak of really high positive things that aren't assembled? I think this might be some sort of redundant sequences being thrown out by the assembler because they're redundant, but I don't know that for a fact. And so we want to go look at these. A couple other things is we actually haven't compared this to any other methods yet. Right? What happens if we just take all 19 million reads and blast them and try to come up with some sort of dividing line and say what happens? Or use, there are other types of ways you can filter the classifiers that we want to look at and see how does this do any better than them. What we did was based on presence and absence of k-mers. Would a frequency-based approach improve specificity? I actually liked presence and absence better because the input data of those genomes was very heavily biased, and I was afraid that frequency-dependent would actually be more biased by the training data, basically, than just using present absence. We actually think these methods might be sort of have some interesting other applications beyond sort of ancient DNA or getting rid of assembly. We actually wonder if these could also be adapted to sorting metadata, genomic data, and that sort of thing. Anyway, we're still developing this. This is done largely in collaboration with Ann Stone and Tom Honap, our student, and then a little more further afield, Jane Bigster, Johannes Krauss, Alexander Erbig, and Kirsten Voss, who are all doing the actual sequencing while I'm helping with bioinformatics again. Thank you. Any questions?
Mike?
Yeah.
Have you tried playing with that with higher k-mer values just to see what happens? Because normally when you do assemblies, you're usually using k-mers when you have performance of thirds.
Yeah, so the the results I actually showed on the real data were I forgot to mention, we're actually for K of 14. I haven't gone a lot higher than that because the code we've written is completely unoptimized, and the memory and speed start to really ratchet up. And so I wanted to do the shortest possible K I thought I could get away with. And 14 worked pretty well. 13 I think would be a disaster. I might try doing just 15 or 16 to see if that makes a